And I'm very happy to introduce our speakers today. Indeed, this is the first webinar where we supposed to have two speakers. Dustin Penn, who has studied the behavior of house mouse and especially wild mice for approximately 30 years. And he received his undergraduate and graduate education in the United States at the universities of Central Oklahoma and University of Florida, and did his PhD in biology at the University of Utah. Dustin has his habilitation from the University of Vienna, and he's accompanied by Dr. Zala, who received her master's from the University of Bern and PhD from the University of Utah in USA. And they together have been researchers at Konrad Lorenz Institute of Ethology since 2002 already. And their research is focused on chemosensory and acoustic signals and sexual selection. And they are interested in adaptive functions and proximate mechanisms of behavior and studying wild and wild-derived mice under laboratory and semi-natural conditions. And today's talk is about courtship vocalizations and how they correlate with copulatory success. So happy to have you, and the screen floor is yours.
Well, thank you, Vootele, for the invitation and for the introduction. Today, I'm going to talk about our work on sexual selection in house mice. So for the first part of the talk, I'm going to give some general background on our work on chemical signals, particularly the stuff we've done in the last 5 years. And then the second half of the talk, I will talk about our work on vocalizations and sexual selection. And in between, since those of you in the audience are focused on home cage monitoring, I will tell you a little bit about our ecological approach to home cage monitoring. So, we study secondary sexual displays, and like Darwin, we're very much interested in how these displays evolve. Despite the fact that they attract predators, Darwin was interested in explaining their benefits, and he realized that if they attract potential mates and can persuade mating partners, they can evolve through sexual selection. But our main question is, do they actually influence mating success? And if so, how does this work and why? Why would females find such elaborate traits to be attractive? So one of the leading ideas for many years is that secondary sexual displays are reliable indicators of male quality. Now, the leading theory to explain why signals would indicate quality is called the handicap principle. We have recently shown that this idea, which is based on a interpretation of theoretical models, and we've proposed different ideas about how this can work. I'm not going to talk about theoretical work today, though. I'm going to talk about our empirical studies on house mice. And so it might seem strange that we would study conspicuous secondary sexual displays in house mice, but most of you know that, contrary to what is often assumed, that mammals lack sexual displays. They, in fact, have a variety of glands, which they produce various pheromones. Males scent mark their territories. This is an example of what scent marks look like under a UV lamp. And so when we're watching the mice in a natural, semi-natural condition, the males spend most of their time depositing scent marks around their territory. These scent marks are highly sexually dimorphic, produced mostly by males and territorial males, and we found that they're indicators of health as well as males' social status, and that they're triggered by when males come across female scent or females, which increases their scent marking and the attractiveness of their marks to females. Now, in the scent marks, urinary scent marks, males deposit a large amount of protein. These major urinary proteins, or MUPs, bind to small volatile pheromones and thereby provide a time release mechanism in their environment to release these volatiles over long periods of time. Without such anchoring, volatile pheromones would volatilize and there would be no scent in a scent mark. So we've studied these signals over many years and found that the first and only evidence, to my knowledge, that scent marking increases male reproductive success, MUP production increases male reproductive success, and also we find correlations with particular pheromones that increase male reproductive success. So male chemical signals are, as and Darwin predicted, the equivalents of male peacock's plumage. And so this provides evidence that they're sexually dimorphic traits maintained by sexual selection. Moreover, chemical signals are extended phenotypes that go way beyond the body. As I said, males cover their territory in these scent marks. And unlike other types of secondary sexual displays, they have action from a distance. And these are very peculiar effects that pheromones have on female behavior and physiology, such as accelerating female puberty and triggering estrous cycling, so-called Vandenberg and Witten effects. We've recently shown that by triggering estrous cycling, that this also enhances males' reproductive success. So now our work has built on studies conducted with laboratory mice, but we don't study laboratory mice very often. Laboratory mice, which we refer to as mus laboratorius, like other people, are rather peculiar animals. They're actually artificial hybrids. Some have referred to them as mus gymnasius, but as I said, we prefer to call them mus laboratorius. They're hybrids from different mus subspecies that have been combined, and these animals have very low, if any, genetic variation, low phenotypic variation, and not just in inbred strains, but in so-called outbred stocks as well. There's often high interstrain variation, so a finding from any one strain makes it impossible to generalize to other strains or wild mice. They often have visual, hearing, and other defects and health problems, and their behavior is actually rather different from wild mus musculus. So for these reasons, to study the behavior of house mice in sexual selection, we study wild house mice. And in Vienna and Eastern Europe, that is mus musculus musculus. There's a hybrid zone that runs down the middle of Europe where musculus and domesticus mate, but as far as we know, it's a stable hybrid zone. So these are the semi-natural enclosures where we study the behavior of not wild mice per se, but usually we trap wild mice and then we raise the first generation or second generation in laboratory and then release them into these semi-natural enclosures where we can observe their behavior with the latest technology that is undergraduate and graduate students and technicians with binoculars. So we identify the mice with unique hole punches in the ears, and we've been doing this for over 2 decades in various studies. Now, it looks like there's partitions inside of the enclosures, but the mice can easily climb over these. We set these up because they provide territorial boundaries that the males defend. And we watch them under red light. And in these conditions, we have provided evidence that males modulate the release of MUPs and pheromones depending on their territorial status, and that these are correlated with males' reproductive success. Now, we also conduct studies in the laboratory, such as this is a Y-maze olfactometer, where we found that females prefer the scent of these territorial males that have high levels of MUP20 and a couple of interesting volatile pheromones. We also conduct studies in smaller enclosures where we can manipulate scent marks, and this is where we found that scent marking is correlated with male reproductive success when females can choose their mates. Now, mice do not rely on chemical signals alone. As many of you probably know, they produce courtship vocalizations, often referred to as song, and these are above the range of human hearing, are somewhere between 20 to 120 kilohertz. These ultrasonic vocalizations were actually discovered in our institute by Wolfgang Scheidt and his student. And I didn't know this until after we already started studying these vocalizations, so just by chance. So over the years, they were studied by several other people, such as Sales and Nyby, but it wasn't until the publication of Holly and Gua that this attracted my attention, because I think, like most people, I assumed that the vocalizations, the ultrasonic vocalizations, were about as interesting as the squeaks that the mice made, which didn't seem very interesting, at least for studying sexual selection. But when we found out that they were producing song, this became much more interesting. It's rather embarrassing to be studying sexual selection in an animal and then just find out that they actually sing like songbirds. So I urge you, if you have a chance, to go online in Holly and Gua's paper, and you can hear these songs when they're slowed down in time and pitch to make them audible, and they sound like songbirds. I tried to do this here, but for some reason it's not working with the Zoom link. So Holly and Gua, they suggested that there are different types of calls that the mice make, and indeed, we verified that and conducted the first spectrographic analyses on ultrasonic vocalizations in wild house mice and confirmed what they found in laboratory mice, and that is the mice produce rather different calls. Now, most of the studies suggested that it was mostly the males that call, and various labs have attempted to record males and females when they're interacting by triangulating the recordings of the different microphones. The most recent study estimates that the males are producing somewhere between 90 to 95% of these vocalizations during opposite sex interactions. So just to give you a brief summary of how we study vocalizations, we first record the mice, but you cannot just simply put a microphone in front of a mouse. It's actually rather difficult to get them to vocalize. And as people found with laboratory mice, the way to get males to vocalize is to present female stimuli, which could be scent, a female mouse, or record them during opposite sex interactions. And then we record the vocalizations and then analyze the spectrograms. And this is just a typical spectrogram showing the change of frequency or pitch of each vocalization over time. And notice this is in very short time zones, that they last less than 0.1 millisecond usually. So they're happening very quickly. So we have an automatic detection method that we use to detect the calls called AMUD, or Automatic Mouse Ultrasound Detector. And then once we detect the calls automatically, we classify these, usually manually, the various types of vocalizations that have been described particularly by Maria Luisa Scattoni and other people. And then we look at the inter-observer reliability, sometimes try to make sure that the people who are the individuals who are classifying them, we look at the consistency between observers. Now, this takes a very long time to process these data, and we estimate that somewhere around 55 hours of recordings, a typical study that we'd conduct requires 300 hours of manual annotation. So what we've tried to do is to develop a method for automatic classification. So this shows you the different types of calls that have been classified in house mice, and Rihanna Abbasi and Peter Baloch have been our collaborators in developing a tool using deep learning, and we published a paper of our tool in which we evaluate the performance compared to the gold standard, the manual classification, and other automated methods. And it looks like our tool is so far has the best performance. However, I must make the caveat that when the mice are recorded during interactions, there's a lot of background noise. And so it's still a major challenge to get any kind of detection or classification working when you have such background noise. Now, to get the mice to vocalize, as I mentioned, we present female odor, and in some of our very first studies, we provided a variety of different female odors to males and found that they vocalize mostly to unfamiliar females compared to the different olfactory stimuli that we offered them. Now, if you offer female odor to male mice, there's an enormous amount of variation in how whether they vocalize or not, and how much they vocalize. And so this just shows you when we start recording a male before presenting the odor and the number of USVs they produce, and then upon presenting odor, you can see there's an enormous amount of variation in how many calls they produce. And if we do this, we repeated this week after week, and you can see there's still a tremendous amount of variation in their calling behavior. And what's interesting is that there's consistency within individuals week to week. And this is true for USV count, the number or rate of calls they produce, and almost everything else that we measure about the male's vocalization. So we then compared the variation within males to between males, and we found that there's greater variation between than within males, and that we can accurately classify individuals with 86% confidence. So essentially, this is the first evidence that the male's vocalizations contain individual signatures, or what's sometimes called voice prints. So we also conduct playback experiments where we take these recordings of mice and we play them back for females, and we look at their ability to discriminate different vocalizations and their attraction to different sounds. And so we have found evidence that they are more attracted to an unfamiliar male than to a familiar male, and they're more attracted to the same species than to another musk species. And females' attraction, though, is rather weak. I must say, they quickly habituate, and they don't really show a very strong attraction. And their attraction, we've more recently found, depends on their social experience and their estrus stage. We can also get males to vocalize by presenting a female on the other side of a divider in what's sometimes called a split cage design. And here you can see the huge number of USVs that males emit. And if you notice, most individuals emit very few USVs at all. And this is true in almost all the studies we conduct. It's rather puzzling that there's such enormous variation and that some males just simply don't vocalize. Now, we've found that as some early experiments with laboratory mice have suggested that we can increase the vocalization of males if we sexually prime them the day before by introducing them to an unfamiliar female. So that will increase the number of calls they make. It increases the length of their calls and the diversity of their USVs, the different syllable types that they produce. So we've also recorded the mice before they can directly interact with the female on the other side of a divider. And then when we remove the divider, record the mice during their interactions. And as you can see here on this graph, there's quite a significant increase of vocalizing, particularly when they're interacting. There's a 5 times increase. The mice also increase the repertoire, the number of syllable types they produce, and the length of these calls. What's most interesting is when we did this study, we presented the males and females with individuals who were close kin, either a sibling or a cousin or an unrelated individuals. And we found that there were aspects of their calls, like the length of their calls, were greater when there were unrelated individuals compared to related individuals. We then bred them, these same individuals, we left them together until they had offspring. And as expected, the unrelated individuals had more litters, greater reproductive success. And to our surprise, there was a difference in the vocalizations and the latency to the first litter, to when they had their first pups. So this is the first evidence in wild house mice that USVs actually influence male reproductive success, and the first evidence that they predict their reproductive success. So in the next study that I'm going to focus on is a study we published recently about the vocalizations of mice during the entire sequence of courtship and mating. Now, the vast majority of studies on the vocalizations of house mice have focused on the very initial interactions when mice come together. There have been a couple of studies that have looked at how they vocalize when they attempt to mount, but there have been no studies to actually describe the vocalizations produced during mating and after mating. And the mating in mice, as most of you probably know, is rather complicated because when males copulate, they can sometimes have 100 copulations before they ejaculate in what is called intermission, for reasons we don't fully understand. And so we're recorded the vocalizations of the mice before, during mating, and after mating, and looked at the changes in vocalizations, the entire sequence. And our second question was whether or not there were any vocalizations that correlate with male copulatory success. So we also wanted to look at the squeaks that the mice make that are partly audible. These are mostly in the inaudible range, and they've been studied by a few researchers who suggest that they're mostly or entirely produced by females, and they seem to be, at least with laboratory mice, associated with repelling males' advances. So we wanted to test this idea as well. So in this study, we recorded the mice during the split cage when we present the males with a female, and then 10 minutes later, we remove the barrier, allow them to interact, record them for another 10 minutes, and then for another 10 minutes, we continue recording in early courtship. But then we waited until a couple of days, recorded them for almost 48 hours, and then examined the videos and the recordings of the mice 10 minutes right before copulation with ejaculation, and then 10 minutes after ejaculation. And we compared their vocalizations and their behavior with the mice that failed to copulate to ejaculation during this time. So this is quite a heroic effort by Therese Klaus, who actually classified 53,000 calls. And we found that 87% of these were USVs and 10% were these squeaks or broadband vocalizations, BBVs. So if you look at this graph, it shows the relationship between the number of USVs and the syllable repertoire, the diversity of calls that the mice make. And we confirmed a previous finding we made that there's a logistic relationship. As the mice increase the number of calls, they increase the repertoire up to the point where they reach the full repertoire. Now, if you notice, there's a tremendous diversity in each recording. They vary from 0 calls to over 1,000 calls per 10 minutes, and the median was around 160 calls per 10 minutes. So this is just a short summary of what we found. Again, back to this repertoire of different call types. And what we found is that in the beginning, while the mice were separated, we found that they're producing these very strange calls that we refer to as unstructured and very short calls. This replicates some other findings we've had previously. And then when they first start interacting, we found again that they produce mostly these very simple types of vocalizations. But then when they start mating in late courtship, and especially during sexual behavior, they produce these very complicated vocalizations with frequency jumps and harmonics. They also have a lot of other differences in the features of their vocalizations. So this is a graph showing the fold changes. This is in the very initial contact and interactions. The mice mostly increase the number of simple calls, and particularly these up-shape vocalizations, whereas in late courtship, they produce more complex calls, and particularly these harmonic vocalizations. And this is now showing you each individual call and their numbers compared to pre-contact, and then during contact, they increased all of the simple calls and a couple of the complex ones. And interestingly, they decreased the emission of these unstructured calls and simple calls. And this is just as cluster analysis to show you the differences between the types of vocalizations they're producing. And then during late courtship, the mice, again, this shows how they increase the production of all of the complex vocalizations. And until after ejaculation, the mice cease vocalizing for up to 5 minutes, and then they resume making vocalizations again, as in the early stages of courtship. So this graph shows the relationship between the number of sexual behaviors the males showed and the number of vocalizations. And what is most interesting is that there's a correlation overall, but particularly for the males that mated successfully until ejaculation. And if we look very closely at the vocalizations produced 5 seconds before an attempted mount or successful mount, the males that mounted successfully produced more vocalizations just before the attempted mount, 2 seconds before the mount. So overall, when we compare the vocalizations of the successfully copulating males and unsuccessful males, we found aspects of the vocalizations that predicted whether the mice were going to copulate or not. And we found correlations with male sexual behavior, particularly among the successful males, and differences in the timing just before the mating attempts. Interestingly, the males that mated successfully also had quieter calls, lower amplitudes than the unsuccessful males. And so overall, we found that the successful males had higher frequencies. They had longer intersyllable intervals within their bouts. And there was a trend for a higher USV repertoire and more complex USVs. So when we looked at these squeaks or BBVs, we found that they were positively correlated with male sexual behavior, though not significant. And they were elevated during the male mating attempts, but this was true whether the mating attempts were successful or unsuccessful. Now, contrary to what we expected, these vocalizations did not repel the males. They did not reduce male courtship, the mount attempts, or their copulatory success. In fact, females often initiated interactions with the males, and the production of these BBVs were positively correlated with these female approaches. Now, perhaps the most interesting result is that the copulatory success was predicted by the timing of the emission of these squeaks, with an increase and then a sudden reduction just before male mounting. And here you can see over time, this is the seconds, once again, right before a mounting attempt, and it shows the mean number of squeaks and how these vocalizations increase up to the mounting attempt and then suddenly decrease 1 second before a mount in the successful copulators. And this is probably the most interesting result of all. This result shows that the ultrasonic vocalizations, which are presumably produced mostly by the males, and these BBVs, which are presumably produced by the females, are highly synchronized. They're emitted at approximately the same time. This spectrogram shows the different USVs. And just below that, you can see these broadband vocalizations, which are highly synchronized. And this actually was first found in a study by Laura Hurley and her students, laboratory mice. And this is the first evidence of this synchrony in wild house mice that happens during, if I remember correctly, they were looking at mounting behavior and not copulation per se. But what we expected is that the synchrony would influence the male's copulatory success, but we didn't find any evidence for that. Okay, so in summary, what I've shown you is evidence that the male's ultrasonic vocalizations during courtship show numerous changes with increased complexity over all phases of courtship and mating, and especially during the male sexual behavior. And these occur right up until ejaculation. And then, as I mentioned, there's a brief pause, and then males resume calling again. This is the first evidence, to our knowledge, that the emission of USVs influence male copulatory success in house mice. And it's not just the emission of certain USVs, but it's their timing that correlates with male copulatory success. I also showed that the squeaks or broadband vocalizations are also highly dynamic, and they're synchronized with the USVs during mating. This is evidence for duetting, as in some songbirds. But these BBVs did not repel the males as we expected, and they correlated with female approaches and timing, which were associated with successful male mount attempts. Okay, so just want to wrap up and tell you a couple of things about our current work and future directions. So I know that most of you are studying house mice in the laboratory and home cage monitoring. So I just want to show you an experiment we published a couple of years ago where we did an experiment in home cages where we introduce male odor or playbacks of male vocalizations or both. And the idea was to test whether or not the vocalizations influence female estrus the way that male odor has been found to do, the so-called Witten effect. And what this graph shows is the number of estrus cycles per female. And so what we found is that if we introduce a male odor daily into the female's cage, that we can induce estrus cycling compared to the control, which had no male odor. But when we introduced the USV playbacks, the recordings of the males, it had no effect on the female cycling, and the combination was really of USV playbacks and odor was not different than odor alone. We then looked to see how that affect the latency for the pair to have offspring, and found that with the odor stimulus that it reduced the latency to the first birth, whereas the USV playback had no effect on the latency to the first litter. So this is the first evidence, to my knowledge anyway, that male scent, the Witten effect, that male scent can trigger estracycline in females in wild mice, but USVs did not have such an effect. And it also provides evidence that the odor increases not only cycling, but male reproductive success. So this is just an example of how with home cage monitoring, not only can record the behavior of the mice, but can actually introduce stimuli to examine how it affects their physiology and behavior. So we have a cooperation with Serena Bovetti and Paolo Pareto at the University of Turin, where we're interested in trying to understand how the brain integrates information from different sensory modalities, in particular auditory, acoustic, and chemosensory modalities. And finally, this is just an example of where we tried to record the mice in semi-natural enclosures, or what you might refer to as ecological home cage monitoring, where you can see we dropped the microphones down into the enclosures to record the mice. Now, the problem with recording mice in such conditions is that ultrasound does not travel very far, and only up to around 1 meter. So it's really difficult to actually capture the mice during their sexual interactions, because they move so far. Typically, when the mice are courting, they will move over a dozen meters or more when they're interacting on a male's territory. So it's extremely difficult to capture their vocalizations. So we have recordings, and we managed to do that by presenting females in cages below the microphones and simply recorded their interactions, and we were analyzing those vocalizations. Okay, so I'd like to acknowledge the work of our technicians, postdocs, and students over the years that performed this work. Especially, I want to say and acknowledge the work of Teresa Klaus and Bettina Vernisch, Eva Strasser, our technicians, and also our PhD students, Maria Adelaide Marconi, Doris Nikolakis, and and Rehanna Abbasi, who really did the work on the vocalizations that I presented. And our other collaborators, whether it's working on models of signaling theory, chemistry, proteomics, bioacoustics, or genetics, we've had a lot of collaborators that helped us to do this work. Okay, so that's all I have. I tried to leave 15 minutes